The gram stain is that differential stain that's going to tell us the difference between an organism with a gram-positive cell wall and a gram-negative cell wall. So it's a process of 4 steps that are going to, at the end, give us our results on which cell wall type that organism has. So we start with crystal violet. Crystal violet is our primary stain. So this is the first thing we apply. After we apply it, if we were to look at these cells under a microscope, both gram-positive organisms and gram-negative organisms would both look purple. So looking at the cell wall itself, or the chemical reactions, we can see that crystal violet is in the peptidoglycan layer, that thick layer of the gram-positive organism. We can see it's loosely attached to that outer membrane, and we can see it inside that thin layer of peptidoglycan in the gram-negative organism. The second step is applying Graham's iodine. Graham's iodine is a mordant. A mordant is a substance we use to lock dyes in to a structure. So when we dye clothes and dye materials, we apply a mordant to cause that cloth to retain that color, retain that stain. Same concept here. That's what we're going to do to these organisms. If we were to look at the cells after applying that mordant under a microscope, they would still both look purple. We've not done anything really to them yet. However, in the cell itself, or in the cell wall, that mordant has caused those dye crystals to be trapped in that peptidoglycan layer. They can't go anywhere. Essentially, we've locked them into place. In that gram-negative, that iodine has absolutely no effect on the crystal violet in that cell wall and cell membrane. So it's not locking that purple into place like it did over here in the gram-positive. The next thing we do, the third step, is apply ethyl alcohol. Ethyl alcohol is a decolorizer, so it's going to do exactly what it says. It's going to wash away that stain. If we, after applying ethyl alcohol, we looked at a microscope, gram-positive cells would still be purple. Gram-negative stains, or negative cells, would be colorless. We wouldn't see a gram-negative organism under the microscope after applying alcohol. So in the gram-positive cell wall, because that mordant locked that crystal violet into that peptidoglycan layer, alcohol has no effect on it. That crystal violet is stuck in place. However, that alcohol in a gram-negative organism weakens that outer membrane. When that outer membrane or that outer cell wall is weakened, the cell loses the dye and washes it away because it wasn't locked into place. It wasn't locked in that peptidoglycan layer. And the last step is counterstaining. It's called safranin. It's a red stain. After we apply safranin, gram-positive cells will still be purple. There's no place for that safranin to stick. Gram-negative cells, however, will stain that pinkish red. So in the cell wall, that red stain, safranin has no effect on a gram-positive. However, that red stain will color that thin layer of peptidoglycan. So when we look at a microscope, we can see gram-negative cells. If we did not counterstain, they would be colorless and we'd see nothing.